but I think we should get started. On behalf of Clinix Health Group and Health IQ Consulting, Dr. Gubega and Dr. Raymond Bila, our clinical manager, and any executives of Clinix Health Group present in the room, I'd like to welcome you all to our webinar. My name is Zinhle Mthembu. I'm the marketing manager at Solomon Sticks Maree Private Hospital and Clinix Botilong Private Hospital. At the moment, I would like to just make a few announcements before I introduce the speaker. All webinars hosted by Clinix Health Group are CPD accredited unless otherwise advised. All personal information is captured at the time of registration, your name, HPCSA or SANC number. It is therefore important to capture the correct information as CPD certificates are auto-generated and the same information you provide when registering will translate into certificates. There is no need to provide your personal details in the chat session tonight. And any errors after the fact may be reported to our admin team via email. The email address is webinars@clinix.co.za. For you to qualify for a CPD certificate, you have to attend the full lecture. You may leave after the lecture, but we find that our most important conversations often take place during the question and answer session at the end of the session. Please stay with us for that. Kindly note that your registration and attendance is monitored through Zoom for the duration of you being online with us tonight. For questions and answers, you may use the chat function to pose questions during the lecture tonight, and the speaker will attend to these at the end of the lecture. We will also allow questions to be posed verbally at the end of the session. For this, kindly use the raise hand function or simply unmute your mic at that time. Would appreciate you keeping your mic muted for the duration of the lecture, and any connection and buffering, poor sound quality issues you should experience, kindly check the settings on your device that you are using, as we will not be able to assist with these remotely. In the event that you received an invitation to tonight's accredited meeting from a colleague or WhatsApp group and you wish to be added to our mailing list for future webinars, please forward your contact details to us. The email address again is webinars@clinix.co.za. At this moment, I would love to introduce our speaker, Dr. Lienna Marie. She is a clinical virologist with Lancet Laboratories. Originally from Pretoria, she obtained her bachelor's degree in medicine and surgery from the University of Pretoria. After her internship and community service training in virology, she obtained her MEd Viral Path Cum Laude at the University of Stellenbosch with a special interest in viral lower respiratory tract infections in children. During her postgraduate training period, she also obtained a diploma in tropical medicine and hygiene from the University of Wits. For 18 months, she was the only clinical virologist in the Eastern Cape before she joined Lancet Laboratories in 2015. Tonight, Dr. Lienna Marie will be giving us an update on the COVID-19 vaccine. Doc, anytime you are ready, please take over.
Thank you, Zinhle. And welcome everyone to join, and thank you for joining me, and thank you to the Clinix group for inviting me to talk. So I'm going to start now, because there's a lot of information that was added in the last week, so this is slightly hot off the presses as much as I could have found that information. Okay. I'm going to make sure that everyone can see my screen. Okay, perfect. Then I move on. So welcome everyone to the talk. Sorry, I just there we go. Okay, so first of all, why do we need and urgently need the vaccine? So almost all populations are still at risk. There haven't been a lot of serosurveys done, but most of them do reveal still large immunity gaps. A meta-analysis was performed towards the end of last year, and in that meta-analysis, the seroprevalence estimate was 3.38%, with a range of 0.37 to 22%. South Africa was not included in that meta-analysis. They only had included a few African countries. The seroprevalence in Kenya, for example, was 5.6%, and the lowest was Malaysia, that's 0.3%, and the highest was Iran at 2%. There was one small serosurvey in South Africa done in the Cape Town Metro antenatal and HIV clinics through the sector, and that showed that the seroprevalence was 40%, with a range between 30 and 46%. Just anecdotal data from Lancet. Our seroprevalence is around 30%, but I mean, that is very anecdotal with a lot of biases. So we all know about COVID fatigue. We're all tired of not seeing our friends and family, and the lockdown restrictions are not sustainable, and no one of us can go at it alone. So this is just a little bit of to remind us how immunity develops. So the virus attaches to the host cell receptor. It then enters your cell. It releases the genetic material, which is then transcribed. So copies are made and translated into proteins. The virus gets assembled and it gets released. So immune response, you have your antigen presenting cells. They engulf the virus and they then present viral particles or peptides to both your B cells and your T cells. Antibodies are produced mainly against the nucleic capsid and the spike proteins, but antibodies that are directed against the receptor binding domain of the spike protein seem to be the main target of neutralizing antibodies. So that is where most, or not all of the, actually all of the vaccines at the moment are pointed towards. So we've got three main categories of types of vaccines: inactivated and live attenuated viral vaccines, then protein subunit and peptide vaccines, and then the vaccines that includes viral vectors and DNA and RNA. So if we look at the different ones specifically, the inactivated one, so this is where the virus is completely inactivated for the safety of the patient, but we need to keep the epitopes intact for the protective immunity to be made by your body. So the problems for that is during that inactivation process, one is very worried about denaturing the proteins, degrading the proteins, and then cross-linking or aggregate formations. It's a very cost-effective way because you just need to grow the virus and then inactivate it either through formalin, ultraviolet light, heat, or formaldehyde. The problem with this one is you still need a BSL-3 laboratory to grow SARS-CoV-2 in. Other viruses may only need a level 4 or 2 laboratory. Then for the live attenuated vaccines, they work very well for acute diseases. The big one, of course, is for MMR, but one is very worried about safety and efficacy with chronic infections like HIV.
Yes.
So what is good about live attenuated vaccine and inactivated, you can produce immune responses.
I keep meeting all of them.
So you can also mass produce, which is good. The problem with this type of vaccine,
because now I activated an option that says mute all participants, and I'm the only one who can allow for them. Yeah,
is contraindicated in immunocompromised patients. So the protein subunit vaccines and peptide vaccines, they only contain specific antigenic fragments.
Okay.
So that eliminates any concerns of incomplete inactivation, virulence recovery, or pre-existing vector immunity, which we will see with the vector vaccines. You can specifically target the well-characterized neutralizing antigenic epitopes. So that's why with COVID, we focus on the spike protein. And most of the time, they are used in combination with adjuvants to improve the immunogenicity and efficacy of the vaccine. So peptide vaccines, those are just smaller amino acid stuck together, is usually less than 50 amino acids, whereas proteins are above 50. So they've got the same problems that you need to add adjuvant, and you can also enhance it by sticking all of your peptides together to make what we call a virus-like particle. Then the vector vaccines, we've got both non-replicating and replicating vector vaccines. So the non-replicating ones, from the name, you can figure out that these virus vectors are not replicating, so they don't make extra copies in your body. What you do is the virus is non-replicating, but it's still alive. So you just insert the gene that you are interested in into the vector. You then inject it into the patient. The viral vector goes into your cells, and then you start producing your antibodies or activate your immune system. The replicating ones, the same principle applies, but this vector can replicate. So the potential challenges, if you think about, especially the replicating ones, that the viral vector will spread throughout the body. You are worried about duration of expression. That is especially with the non-replicating ones. You can only you only give so much virus, and then it's sort of broken down. People are always worried about chromosomal integration, but that has not been proven yet, especially with the RNA vector vaccines. I mean, RNA cannot be integrated into your DNA. And the biggest problem with these types of vaccine is if you have pre-existing immunity against the vector, because then your body will recognize the vector as soon as it enters, and then you can start producing a robust immune response before the vector can actually produce the spike protein for your immune response to react to that. Then the nucleic acid vaccine. So these are the new kids on the block. We've got DNA-based and RNA-based. So with the DNA-based, the viral genes are in the form of DNA plasmids. They are then given via intramuscular injection, either of the naked plasmid, or it needs to be encased in a liposome or some nanoparticle to not be degraded in your body as soon as it enters. So in these, we sometimes also have to add that polysaccharide adjuvant to enhance your immunity. So the DNA vector one, once it enters the cell, it actually still needs to go into the nucleus to then use your host cell machinery to make mRNA from, so that you can produce the proteins and show it to the rest of your immune system. So no DNA-based vaccines have been approved as yet, but the good thing, we are worried about integration, but the clinical trials so far on any DNA-based vaccine has showed that there's no chromosomal integration, nor is there development of anti-DNA antibodies, so you don't need to worry about those pre-existing immunity like you need to worry with the vector-based vaccines. So they are used as a prime boost strategy with those vector-based vaccines, so you do not have that pre-existing immunity with either one of them. And it's also scalable for mass production. So RNA-based, so these are the new ones with like Moderna and Pfizer we have now. So that contains selected viral genes in the form of messenger RNA. It also needs to be coated with lipids to enhance uptake. And these, because they're already mRNA, they are immediately translated in the cytoplasm of the cell. They do not enter the nucleus at all. The RNA, you can see, is directly here. So the disadvantages of this is that mRNA is highly sensitive to degradation because it is a single-stranded structure and you've got RNases in your cells. So you cannot make any additional mRNA copies, and that cannot be incorporated into your DNA because it never enters the nucleus of your cell. So to improve stability, they've added the lipid nanoparticle. They try to modify some of the nucleotides, and they also have a process which they call sequence optimization. So this is also rapidly scalable, cell-free manufacturing, and as I mentioned, no risk of chromosomal integration. One company is looking at using mRNAs that do include the replication machinery of some viruses so that you can actually generate mRNA so that you can give lower doses and get a better immune response, but that is still sort of in the trial phase, not in humans as yet. So just to remind us about the different types of clinical trials before a vaccine is approved, we have our preclinical testing. So that's in vitro and animal studies. You try to identify appropriate dosages, pharmacokinetics, and toxicology. Then we start with human testing. So phase 1 is you look for safety, dosage, and the capacity to induce an immune response. So it's a small number of patients who are completely healthy. Then phase 2, you further look at safety and efficacy in a larger group, so a few hundred people. And then phase 3 is more definitive data on efficacy. So these are the large randomized blinded controlled studies where you include hundreds to thousands of patients. And once it's approved, there's always phase 4 if there are a few additional safety or efficacy concerns. And then after introduction, you also have oversight. So if anyone develops an adverse event or a reaction to a vaccine, that is reported and it is investigated to see what is going on. So this is just to explain visually how that happens. So we have the different phases that follow one another sequentially. So you can only imagine it takes several years for a vaccine to be approved. So if we look at a few examples, poliovirus took 47 years, rotavirus 33 years, human papillomavirus 22 years at least, and then of course HIV is still elusive. So this is what authorities have done for coronavirus due to the pandemic. We can't wait 10 to 15 years for the vaccines to roll out. So they've come up with this staggered approach that each phase sort of overlaps with the previous one, and they've got certain set points to say, yes, if that part of phase 2 is, even though it's not complete, if that is successful with these sort of stopping points, or just not stopping points, but interim points, then we can start the other phases. So this is very helpful for the mRNA-based vaccines, vectors, or recombinants, because we only need the genome sequence of the target, the spike protein, and we can make it, and we're not working with any infectious SARS-CoV-2. It's safer because we don't modify the human genome and we don't infect the recipient. We don't use weakened or inactivated virus. So we've gone through the process much quicker. So if you look at the vaccines that are available, this is from the Washington Post. So this is data from the US. So they have only approved Moderna and Pfizer to date, whereas other countries have approved more vaccines. So 11 vaccines have overall have been approved worldwide. We are not going to look at all 11. That will take forever. So we're going to look at sort of the top 5, actually 6 now, but what is potentially going to be available in South Africa. So if we start with Moderna, So that is a lipid nanoparticle encapsulated mRNA vaccine and encodes the spike protein. So if you look at the phase 1 study, so that was just dose escalation, trying to find out, as we've mentioned, very few patients were all healthy adults. So we looked at these different doses, and all of the patients had immune responses adequately. So if you can see on the graph here, these are the different doses, and over time, what happened to their immune responses. So this is ELISA antibodies, or antibodies we detected via ELISA against the spike protein, and this is a virus or pseudovirus neutralization assay. And here on this side, we can see there are the comparison of the convalescent sera. So in all of the pictures I'm going to show, most of them have that convalescent on the right-hand side. You can always compare to that. Just continue to move something. There we go. So the adverse events in the patients were fatigue, chills, headache, myalgia, pain at injection site, the normal side effects we see after most vaccination. There were no serious adverse events. One participant in the 25 microgram group withdrew due to an unsolicited adverse event of urticaria, but no one else did. So Moderna decided to use the 100 microgram dosage to take forward with the other studies, and antibody levels remained elevated up to 119 days in this group. Mention a phase 2 where they had 600 where they saw 50 micrograms.
I never studied them.
But they haven't posted any results. So this is the phase 3 randomized and a 1-to-1 ratio placebo-controlled
boarding school
with over 30,000 participants.
Boarding school, you stay at the school.
All adults, the mean age was 51.4 years. They were all in the US. So 97% of patients were white, but they did mention that the racial and ethnic proportions in the study were representative of US demographics. So they were sort of really skewed towards the US. So they included 24% of the patients were over 65 and 16% of the under 65 had comorbidities. And you can see it was almost half of patients were females. So they used the 100 microgram dose, and it was 2 doses, 28 days apart. And primary endpoint was prevention of symptomatic disease at least 14 days after the second injection. And then further, they looked at prevention of severe disease and prevention of infection by SARS-CoV-2 with any symptom. So these were the results they showed with a lot of fanfare towards the end of last year. So we've got there were 196 cases of COVID-19 in the study, with 185 of those in the placebo group. So that gave us that efficacy of 94.1%. In the study, there were only 30 cases of severe COVID, and they were all in the placebo group. So efficacy against severe COVID was 100%. And efficacy was also consistent across both age, race, and ethnicity, and gender. And they had the same solicitous adverse events: pain at injection site, fatigue, myalgia, headache, and redness at injection site as well. When they looked at efficacy 14 days after the first dose, that was, sorry, I'm trying to move them, can't move over to that. Oh, wait, here we go. Apologies. Was 95%. The problem I had with this study, they only tested symptomatic individuals. So they didn't at all look at prevention of asymptomatic infection. And as we've all seen, a lot of patients actually don't have any symptoms who have COVID. There was only one COVID-related death, and that occurred in the placebo group. And they've mentioned that they are starting testing in adolescents between sort of 12 and 17 years of age. So Moderna has been approved in 37 countries worldwide. So if we look at Pfizer-BioNTech, so in the phase 3 study, it was also a placebo-controlled dose escalation study. They used 2 candidates. So the same lipid nanoparticle nucleoside modified mRNA. So the first one only encoded the receptor binding domain. The second one encoded the entire stabilized prefusion spike protein. So very similar to the Moderna, this one. So phase 1, they included 195 participants, all older than 18 years of age. And as we can see, there were quite similar antibody responses between the different groups and the different doses as well. So local reactions was once again pain at injection sites, and these local reactions were more frequent after the second dose. And systemic reactions patients reported included fever, chills, fatigue, and headache, and also greater after the second dose. Interestingly enough, the side effects were, or adverse reactions were lower in the older patient group, and most of these symptoms generally started day 2 after the injection and then resolved by day 7. So we can see there were similar antibody responses. So this is the dosing they decided to take forward, and they also decided to take forward the fully stabilized spike protein due to the lower incidence and severity of systemic reactions. So in their phase 2-3 trial, so 1-to-1 ratio, more than 40,000 participants older than 16 years of age in 6 countries, which did include South Africa. So they used the 30 microgram dose given 21 days apart, so that's 3 weeks apart. The majority of the patients were in the US, and 42% of them were older than 55 years of age. The primary endpoint was prevention of confirmed COVID-19 at least 7 days after the second dose, and then they also looked at prevention of severe disease. So in this study, there were 170 cases, 162 in placebo versus 8 in the vaccine group. So that gave us an efficacy of 95%. So there were 10 cases of severe COVID after the first dose, and 1 was in the vaccine group. They didn't mention exactly if that one was how long after the first dose or if it was prior to the second dose. Same local reactions, mild to moderate pain at injection site was the most common one, and once again, less frequently in participants who are older than 55 years of age. Same fatigue, headache, fever, chills, also younger group, second dose. Adverse events was 1 case of lymphadenopathy, and 4 cases of vaccine-related serious adverse events were mentioned. So that was 1 patient who had a shoulder injury, the axillary lymphadenopathy, 1 case of paroxysmal ventricular tachycardia, and 1 case of right leg paresthesia. But there were no COVID-related deaths in this study. So this vaccine has been approved in 55 countries worldwide. So if you moved over to Oxford Astrozeneca, so that's the one that has arrived in country. This is a replication deficient chimpanzee adenovirus vector. So it's a different type of vaccine, but it also encodes the spike protein. So in the phase 1 study, single-blinded randomized controlled trial, just over 1,000 participants in the UK. So this was quite an interesting study where they actually had 3 different groups. So they had a single dose of the vaccine. Their placebo wasn't actually a placebo. They used the meningococcal meningitis vaccine because they knew the vaccine would cause sort of the local side effects of pain at injection site and redness and all of that. So they wanted to create that same local side effects in the placebo group, so that, because they didn't want to tell people which vaccine they got. So they gave a vaccine that they knew would cause exactly the same type of local side effects. And then in 10%, not 10%, 10 patients, they used a prime boost strategy where they gave them a second dose of the vaccine after 28 days. So the median age in this group was 35 years and 90% of the participants were white. So local and systemic reactions were more common in the vaccine group, but they were less severe after the booster dose. At two of the sites, they halfway through the study, they allowed patients to actually take prophylactic paracetamol and to continue that for 24 hours after the injection. And that helped to reduce the commonly reported local reactions of pain at injection site, fatigue, headache, myalgia, and malaise. There was one case of a severe reaction in the study, but that was actually in the meningococcal meningitis group, where one patient had hemolytic anemia. So we can see that sorry, I didn't show that, but they say that the specific T-cell responses peaked on day 14, and this is for IgG or responses, and they peaked by day 28, and we can see the boost here at around day 28, where we gave the booster dose. So that is why they decided to move on with the prime boost dose, because the antibodies remained elevated up to day 56 at least, and they were much higher in this group. So that's why they decided to use that for the phase 3 trials. So if we look at the phase 3 trials, so it's still ongoing, but they sort of gave the final primary analysis on Monday. So that's 4 blinded randomized controlled trials in the UK, Brazil, and South Africa. So in this analysis, they included just over 17,000 participants. They were all older than 18 years of age. So we can see the majority were in the UK. So unfortunately, they didn't give much demographic information in this latest update that they released on Monday, which included the South African data. The first publication, this December one, was only the UK and Brazil data. So these demographics don't include the South African data, unfortunately. So in this study, we actually had 3 groups: a standard dose group, a low dose standard dose group, and a control group. So in the control group, they either gave saline or the meningococcal meningitis vaccine. So in the UK, they gave 2 doses of this, the meningococcal meningitis vaccine. In Brazil, they gave meningococcal meningitis and saline, and in South Africa, they gave 2 doses of saline. So this low dose standard dose group was an interesting one that sort of arose by chance, because they used different manufacturers to create the vaccine for them. And at that stage, there were different sort of quantification methods to make sure that there was enough virus in the vaccines. And everyone thought the 2 main methods of spectrophotometry versus quantitative PCR were equivalent. So they said, you know, the manufacturers could use either one. But then later on, they realized there was actually a marked difference between the 2. And that's why they then sort of, there was a lag in the study. And they were worried about this group, the low dose standard dose group, that they sort of got way too little vaccine dose in that first one. So they sort of halted the trial at that stage and looked at all of the data. But when they looked at just the antibody levels produced in that group versus the standard dose group, they saw that they were similar, so that they decided to continue with the primary efficacy analysis. Was symptomatic infection 18 days after the second dose. And the interesting bit was in the UK study. They also asked the patient weekly to do a self-administered nose and throat swab because they wanted to assess asymptomatic infection as well. So just a little side note, due to this sort of halt while they were looking at the low dose group, at least half of the patients in this low dose group and the amount of the standard dose group, they didn't specify the percentage, actually received their second dose more than 12 weeks after the first one, and not sort of the 3 weeks that they initially said would need to be done. So if we look at the start of the results, in the sort of analysis that was released on Monday, they said there were 332 cases of symptomatic COVID-19, so 84 in the vaccine group, 248 in the control group. So that gave an overall efficacy of 66.7%. So if they broke it down between the standard dose and the low dose group, we can see that there is a difference between the two. So they were sort of thinking about, is that difference due to the dosing or is it due to something else? So that's where this data comes in, the interesting stuff. So when they looked also at the asymptomatic infection, the efficacy wasn't brilliant. It was 22%. And once again, we can see that marked difference in efficacy between standard dose and low dose group. So they were thinking about it again, is it dosing, is it interval? And when they did more analyses or breakdown of the data, they found actually that most of this increases in efficacy was actually due to the increase in interval between the doses and not actually the doses themselves. So if you just look at symptomatic patients and there we go. I think everyone can hear me again. And if you just look at the asymptomatic cases, you can see there was it was not efficacious at all if the interval was less than 6 weeks, but it was 47% efficacious if the doses were more than 12 weeks apart. So this is sort of the data why the UK government has gone and said, you know what, you can give the doses between 3 and 12 weeks apart, and a lot of them are trying to stretch it out to 12 weeks because of the data that seems to indicate that an increase in the interval does increase the efficacy. So in the study, 24 patients were hospitalized, only 2 in the vaccine group and 22 in the placebo group. There were 3 cases of severe COVID-19. In the initial study, they mentioned only 2, and both of them were in the control group. Unfortunately, in the preprint study that came out on Monday, they didn't mention in which group that 3rd case was. And that 1 patient died. I don't think they mentioned that either, unfortunately. So this was another halt in the study when 3 cases of transverse myelitis were reported during the study, 2 in the vaccine group, but only 1 they felt was possibly related to the study, not the others. So important thing to remember that Oxford then has asked the Serum Institute of India to also produce this vaccine for them. And that is the one that is called the Covishield. Sorry, no, can't. There we go. And that's the one we have is approved here, and it has arrived here. So the Oxford AstraZeneca has been approved in 49 countries. So if we move on to the Johnson & Johnson vaccine, so that is also a replication incompetent or deficient adenovirus 26 vector that expresses full-length and stabilized spike protein. So phase 1/2A interim results is from Belgium and US. So older patients, just over 400 patients, most white. Oh, sorry, in this trial they actually had 3 cohorts. So that was the younger age cohort. They had a longer term cohort, which is still ongoing, and they haven't released any data. And then the third cohort is the older patients, also majority white. And in each cohort, they had 5 groups where they looked at low dose, placebo, high dose, placebo, and placebo. And they once again were just looking at safety, immunogenicity. So if we looked at the results from this phase 1 trial, solicited adverse events, the normal, what we see, oh, sorry, can you see? Seems the pointer has gone. Apologies for that. Let's see if I can get it back. Ah, there we go. So the same, fatigue, headache, myalgia, injection site pain, low-grade fever, so there were no grade 4 events, and all fever cases were in the first 2 days after immunization, and they resolved within 1 to 2 days. The adverse events were less common in the cohort 3, so that's the older cohort, than in 1, and less common in people who received the low dose regimen than the high dose one. Reactogenicity was also lower after the second dose, so similar to the Oxford one, which is, and then different from the mRNA vaccines, where the patients had more adverse events after the second dose. So there were 5 serious adverse events listed. 1 patient with hypertension that had a history of blood pressure problems, 1 patient with bilateral kidney stones that had a history, 1 case of Legionella pneumonia, 1 case of worsening multiple sclerosis in a patient that actually they were able to prove had, even though it wasn't diagnosed at that stage, they had MRI images of the patient for several years prior to the study, and they could see sort of radiological evidence of multiple sclerosis in that initial scan as well. And then 1 case of fever where the patient had to be hospitalized because they were suspected to have COVID. So that occurred within 1 to 2 days after the first dose, but the patient recovered within 12 hours and it wasn't COVID. So none of the others that I've mentioned were deemed to be related to the vaccine. So antibody titers increased and stabilized by day 71 after the first dose. So they continued to their phase 3 trial. So once again, randomized, double-blind, placebo-controlled, still ongoing in the US, Latin America, and South Africa. So they are using a single dose, which is a bonus point. It is stable at -20 and it, for 2 years and at least 3 months at refrigerator temperatures. So these studies included adults older than 18 years of age, over 40,000 of them. 34% of the participants were older than 60 years of age. They included all ages, and 41% of the patients had comorbidities. So these are the results. They haven't been published yet. This is just from their sort of press release during the beginning of the week. So overall efficacy of preventing moderate to severe COVID-19 28 days after vaccination was 66%. So that is markedly less than what we saw with the mRNA vaccines. So it was 72% in the US, 66% in Latin America, and 57% in South Africa. Importantly, efficacy against preventing severe COVID was 85%. And most importantly, there were no cases who required medical intervention in the vaccine group more than 28 days after the vaccine. So the vaccine was generally well tolerated. Serious adverse events were actually higher in the placebo group. There were no cases of anaphylaxis. And the protection was generally consistent across race, age, and variants. They found that 95% of the cases in South Africa were the new 501YV2 variant. So Johnson & Johnson has submitted their application to SAHPRA for approval. They said they will imminently also submit to the FDA and I presume the European guideline community as well. And this, of course, is also the vaccine that Aspen Pharmacare in Port Elizabeth will help to do the final full contract for them. But it has not been approved in any country as yet. Next one is the Novovax vaccine, which they also gave out new data at the beginning of the week. So this is just a spike protein-based vaccine. So it's just the protein subunit. So it's analogous to, for example, the hepatitis B vaccine, which is also just a protein subunit. Interesting enough, they use a baculovirus to deliver the genetic sequence of the spike protein to full armyworm moth cell line, and the moth cells then produce the spike proteins. The spike proteins are then harvested, if I can call it like that, and they are started onto a synthetic particle, and that synthetic particle together with an adjuvant called matrix M is then injected into the patients. So it's also 2 injections 3 weeks apart, and phase 1 data showed that it was well tolerated with mild reactogenicity, and there were 100% neutralizing antibodies after the second dose. So this is the results that came out at the beginning of the week, also with a press release. So in the UK phase 3 clinical trial, they had more than 15,000 participants. 27% were older than 65, and the primary endpoint was PCR confirmed symptomatic infection at least 7 days after the first dose. So the interim analysis result, there were 62 cases, 56 in the placebo group, 6 in the vaccine group, and 32 of these 62 cases were the new variant, UK variant, if I can call it that. So the overall vaccine efficacy was 89%, which is really good. And there were 61 cases of mild to moderate infection, and only 1 of these 62 cases was severe, and that was in the placebo group. And they also mentioned that adverse events were balanced between the 2 groups. They didn't mention exactly what those were. Then the South African data was a phase 2B clinical trial. They included 4,422 participants. 245 of them were medically stable HIV positive patients. And this is the data from the South African trial. So overall vaccine efficacy was 49.4%. They only looked at the HIV negative group. There were 29 cases of COVID-19 in the placebo group, 15 in the vaccine group. And preliminary data once again showed that the cases were our local South African variant. Only 1 case of severe COVID, and that was in the placebo group. Unfortunately, they didn't give out much information, so we are waiting with bated breath the data for the data to be published, and this has also not been approved. Okay, so this is just a little summary of the vaccine technologies, efficacy, dosages. So only Johnson & Johnson is a single dose. All of the others are 2 doses, either 4 or 3 weeks apart. Oxford AstraZeneca give 3 problems we have with the mRNA ones are the -70 here. I mean, not a lot of places have that refrigerator capacity, and then -20 for these, and the others you can go for several months to years at lower doses. So just a little bit about the Sputnik V vaccine. So that's the Russian vaccine. So that's a heterologous replication deficient adenovirus. So they use 2 different adenoviruses, adeno 26 and 5, to get past that vector immunity that if you give a booster dose of the same virus, you may actually have a poor response. Also expressing the spike protein. So also randomized, double-blind, placebo-controlled. The interesting thing is the ratio they used was 3:1. So they had 3 times the number of people in the vaccine group than in the placebo group. So this is the interim results from their study. Older patients, majority male, majority white, 24% had comorbidities, and they looked at confirmed COVID-19 at least 21 days after the first dose. So their results, we can see overall efficacy was 91%, 20 cases of moderate or severe COVID-19, all in the placebo group. Same adverse events. So the numbers of serious adverse events, that's a big group, but the percentage you can see is kind of the same, and that is reflected due to the ratio that they used. And they deemed none of these to be vaccine related. There were 4 deaths, 3 in the vaccine group and 1 in the placebo group. Once again, they said none were vaccine related. So the deaths were in the vaccine group. 1 was a patient had a thoracic vertebral fracture, and 2 of the other patients actually developed COVID-19 and died due to that. But the symptoms of both of those patients started on day 4 and day 5, respectively, after the first dose of the vaccine. So they said that they think the patients actually had COVID when they were vaccinated, despite having a negative PCR at that stage. And it wasn't due to the vaccine. And the patient in the placebo group had a hemorrhagic stroke. And this vaccine has been approved in 16 countries. I'll try to move a little bit faster with the next little bit. So if we just looked at the percentage of country population covered under the vaccine contracts, you can see a lot of countries have covered more than their population, whereas South Africa, we're probably only around 5%. It might be higher now after President Ramaphosa's speech on Monday. This data I pulled out last week. So cost of the vaccines are the big stumbling block, I think, in our country and all other African countries. So based on the cost we see here of the Astrozeneca, that can explain why AstraZeneca has the highest number of ordered vaccine doses in relation to all of the others. So if we look at the number of vaccine doses that have been administered up to the end of last week, most of them have been administered in the US and China. But if you look at the proportion of people in each country, Israel is leagues ahead of the other countries, where they've vaccinated 45 out of 100, so about 45% of cases. And we go down to all of them, we're not even here. And the US is 7.5%. I think they're a little bit higher now. So if we look at the 5 key considerations in choosing the most appropriate vaccine in South Africa, we need to look at safety profile, in all different patient groups, pregnancy, immunocompromised. Efficacy, is that efficacy similar across all populations included in the trial? So that's why most of the trials do make a point of being more diverse in the participant demographics that they include. Cost is always a big issue in South Africa. Ease of storage and administration, -70 is the problem with Pfizer, and the number of doses is also important. So ideally, we would actually like the single dose vaccines. So just a little bit about how Covax works. So that's a coalition led by WHO, the Global Alliance for Vaccines and Immunization, and the Coalition for Epidemic Preparedness Innovations. So the facility is designed to guarantee rapid, fair, and equitable access to vaccines for every country in the world. So that is by sharing the risk associated with the development, investing in manufacturing upfront, so the vaccines can be deployed at scale as soon as they are successful, and then pooling procurement and purchasing power. And the goal of Covax is to obtain and deliver 2 billion doses by the end of 2021. The problem for South Africa is that we are not one of the 92 economies that will be supported by the advanced market commitment. So if we partake in the facility, we pay the same way as the other countries, for example, Ireland, Japan, Monaco, New Zealand, Switzerland, to help cross-subsidize these 92 economies. So to help with this pricing problem, both South Africa and India have called on the World Trade Organization to suspend intellectual property rights related to anything related to COVID-19. So that's not only the vaccine, but also testing, sort of the test kits themselves, ventilators, masks, anything that is needed to do this. But yeah, I'm not sure if the World Trade Organization will listen to the two of us. So these are the consignments that President Ramaphosa mentioned on Monday during his family meeting about what the government has procured for the country. So these are the 1 million doses of the Covishield, so the Oxford Astrozeneca vaccine that arrived on Monday, a further 500,000 doses, another 12 million doses through Covax, 9 million doses of Johnson & Johnson. Pfizer has committed to 20 million doses, and then they mention also additional doses through the African Vaccine Acquisition Task Team. Didn't mention exactly how many doses. And then just sort of identification and prioritization of target population by the government. So we don't have enough for everyone at all immediately. So it was decided to prioritize people to be essential for social functioning, those most at risk of infection and serious outcomes. So that includes patients over the age of 60 years, those with comorbid conditions, those living in overcrowded settings, and then most at risk of transmitting. So that's where the 3 phases from the government comes from. Limited doses available, phase 1, that's where we are now, and then phase 2 and phase 3. So phase 1, healthcare workers, target population of 1.25 million, then other essential workers, people in congregate settings, so that includes old age homes, prisons, things like that, people older than 60 years of age, and people with comorbidities, and then in phase 3, it will be the rest of the population. So just a last little bit about myths and facts about the vaccine. I'm sure you've received a lot of these things from your friends and family in the last weeks, and this is the important thing we need to try to get over the myths or just because the Ipsos surveys showed that only 47 to 64% of South Africans would accept the COVID-19 vaccine. And if we need herd immunity of at least 67%, we have our work cut out for us. So the first myth is that the vaccine isn't safe because it was made in a hurry and hasn't been properly tested. So there has been research into coronaviruses for several years. So based on that back knowledge, we could apply that knowledge to create the COVID vaccines. And then, as I've shown you in the other slides, hundreds of thousands of people have taken part in well-conducted and carefully supervised trials in many countries, including South Africa. Next myth, I've already had COVID-19, so there's no need for me to have the vaccine. So antibodies are made during natural infection, disappear within a few months, and being vaccinated will boost these antibodies and ensure that protection lasts for longer. I know a few of the sort of guide communities or societies overseas, due to the constraints we have in vaccine supply, do tell people that if you've had COVID and you feel that you are willing to wait for more doses to become available, and you would rather donate your dose to, you know, an elderly person or someone else to get the dose, they say it is possible. So that is something to consider if you are willing to take that chance. The next one, there's a microchip in the vaccine. So that's a myth. So the content of every vaccine has to be declared, and this is already checked by national reference laboratories. So that is what's happening at the moment with the first consignment. That's why we haven't started vaccinating people as yet. And if there's anything out of the ordinary, we would definitely detect it and not use it. 5G mobile networks spread COVID-19. So this is not a computer virus. It is a virus that infects humans and animals, and that cannot travel on radio waves or along electronic networks. So, and the epidemics have also occurred in many parts of the world where there is no 5G. The vaccine will affect your DNA. I'm hoping, from what I've shown you, that none of the vaccines can contain any DNA, and even the messenger RNA ones. Messenger RNA is not DNA, and messenger RNA is broken down quite quickly in your cell. It only contains a fragment of the virus, whether that is the RNA or the protein itself. You give it into a muscle, you stimulate the immune response, and then the fragment is broken down. Next one, getting the vaccine means I can stop wearing my mask and taking other precautions. So if you get vaccinated, you still need to practice prevention precautions. They do not stop SARS-CoV from entering your body. They only prevent you from developing moderate to severe disease. It's not yet clear if patients vaccinated can still carry and transmit the virus, even when they themselves don't get sick. The side effects of the vaccine are dangerous. So yes, there are side effects. None of the vaccines in the world, whether it's for COVID or anything else, is not without side effects, but the vast majority of them are short-term and not serious or dangerous. It's important to explain to people that, you know, the pain at injection site, the body aches, the headaches, the fever lasts a day or two, and they are just signs to show that the vaccine is working and stimulating your immune response. So there was just recently data from the CDC. People were worried about anaphylaxis after the Pfizer vaccine, and they showed that there were 21 cases of anaphylaxis after more than 1.9 million doses of the Pfizer vaccine administered. That gave an incidence of 11 cases per million doses. And just to sort of compare or juxtapose it to the chance of developing long COVID, which at the moment seems to be between 10 and 15%. I don't think I would like to have long COVID. The last myth is the vaccine can affect women's fertility. So it will not affect your fertility. This confusion came through a false report surfacing on social media to say that the spike protein of the virus is the same as another spike protein that is involved in the growth and attachment of the placenta during pregnancy. That is completely false. There are two different spike proteins entirely. It will not affect any woman's fertility. And getting the vaccine will not affect your fertility at all, even if you need to get IVF. And of course, getting COVID, on the other hand, can have a serious impact on pregnancy and the mother's health. Then the last little thing, it's important to remember that the vaccine should not be used on its own. It should form part of what they call the Swiss cheese model of pandemic defense. So every single layer has to be used together, because all of them have holes and multiple layers will improve success. So we need physical distancing, hand hygiene, not overcrowding, good ventilation, isolation, vaccines, test and treat, avoid touching your face, mask. All of that is needed to decrease the chance of you being exposed to the virus and getting infected to the minimum. And that is the end of my presentation. Thank you so much for listening to a very long talk of, unfortunately, over an hour. Let me stop sharing the screen and then we can look at the questions people have. Sorry, I'm just trying to figure out how to stop. Oh, sorry, stop share. There we go. Okay. Okay, I'm actually not too sure how to read the questions. Let me go onto chat first. See what I can see here. So, sorry, let me go to the top. Oh, we've got a lot of questions. Let's see how many we can start with. Sorry, sound. Sorry, I'm trying to find a question. If anyone has a verbal question at the moment while I'm reading through these, let's see if anyone has another question. Okay. AK Desai asked, is it true that if AstraZeneca vaccine is followed by Pfizer 28 days apart, it will be more effective and give better immunity? We don't know that yet. That is actually something that the UK government is looking into. I'm not sure if it's AstraZeneca first and Pfizer second or the other way around, but a few countries or studies are looking into using different vaccines to see what the effect is on immunity with that. So we don't know. Theoretically, using two different vaccines shouldn't be detrimental, but all of this still needs to be confirmed. So will there be decreased immune response efficacy if the booster dose are taken more than 28 days after initial vaccination? I think with the AstraZeneca, it seems with this vaccine specifically, taking it later, 3 months after the first dose, seems to have a better response if you increase the dosing interval between the two doses. So what is advisable if known about how long one should wait between having the flu vaccine and the COVID vaccine? Excellent question. I think what I've saw the latest data from the UK, they suggested waiting a month between the two, but there is no hard data. Trying to think, did they say they're actually going to investigate that? I think so. Don't take my word for it. But I think with most vaccines, we do say if you've not proven that they can be used concurrently, it's better to wait at least a month between them. Okay. So does the initial and booster dose of vaccines have to be the same type? I think we've tried to answer that. So a lot of the times they don't actually need to be the same type. It's better to have two different types. I know after the initial data from AstraZeneca came out, the Gamaleya Institute from Russia actually suggested that they should collaborate in a different study to use the AstraZeneca as a prime and then boost with one of the Gamaleya Institute or the Sputnik doses to see if that gives a good response. They said they were interested, but I haven't seen any data from to say that they are doing it. Then I do see I've got a raised hand from Sonke Dlamini. I'm not too sure if I can unmute you, Sonke. Otherwise, you can just unmute yourself and ask your question. Sorry, I can't seem to. Zintle, will you be able to unmute them?
Yes, Doc, apologies there. I got kicked out of the meeting, so all the questions.
Oh, actually. Okay. Seems Zintle is stuck. I'm not sure if I'm stuck. I hope not.
Oh, okay. Let me ask that question. The next 10 years, that's not indicated for kids.
Sorry, Zintle, you broke up a little bit.
Kids with comorbidities, or persons older than 16 or 18. Are you there? You seem to be trying to sort it out. Are you with me, Doc?
I can hear you now.
Did you get my question?
Not really. Would you mind repeating it again?
Okay. Ooh.
Wants to know about kids under 16 with comorbidities.
Oh, okay. Sorry, colleague said he heard the question. He said he thinks it's about children under 16 with comorbidities. Unfortunately, we don't have any data on that, so I cannot definitively say anything about that.
That's not. Are you there, Doc? And sort it out. Are you with me, Doc? Did you get my question? Are you there, Doc?
Not really. I heard comorbidities. I wasn't sure if the patients need to be less than 16 years or older than 16 years.
Dr. Maree, can you hear me?
Yes, I can.
I think we're having some technical problems, and Zintle is struggling with her line.
Yes.
So for now, what I'm going to ask is that if anyone has a question, like Dr. Sonke has his hand up, to unmute themselves and ask the question. Otherwise, we can just go through the questions on the chat. I don't know if you'd like to assist you with that.
Yeah, I can continue with that, but Dr. Dlamini can definitely unmute himself and ask the question.
Okay. I think let's give him a few seconds to unmute himself.
Okay.
And if he struggles, then we just go on with the questions on the chat. I think he has unmuted himself. Thank you.
Thank you. Sorry, Dr. Dlamini, you are muted again. Okay. Hi. Yes.
Hi. Hello.
Yes, I can hear you, Dr. Dlamini.
Yeah, there was a vaccine that if you receive, it's not actually a vaccine, it's a genetic treatment. And that if you receive this vaccine, your body actually becomes a GMO, a genetically modified organism, in the sense that the body accepts the genetic material and starts producing the spike proteins. So according to her, it is not a vaccine to that extent, but a genetic modification.
No, we're not modifying your own genes. We're only giving you messenger RNA that is broken down quite rapidly. It never goes into the nucleus of your cells. It is never reverse transcribed into DNA. It doesn't go anywhere near your genes to modify them at all. No.
Okay. Then there were some videos that show patients that develop a syndrome that's like a tick with all sorts of abnormal movements in the throughout the body and especially on the tongue. Is there any truth in that?
I must confess, I've not seen those videos and I haven't heard any reports about abnormal movements in people who have been vaccinated. I think that would warrant further investigation to see exactly, you know, what vaccine was given, how soon afterwards, and if they can identify what could have precipitated any of these abnormal movements. So unfortunately, I won't be able to answer that question, but I'm sure there are researchers and the vaccine manufacturers and government will definitely investigate anything that is reported that is abnormal like that. So I think that is the important thing to take home from that is in this case and with any vaccine, if a patient develops side effects or symptoms or something soon after the vaccine is administered and you are worried that it could be vaccine related, you would actually need to report it so that it can be investigated to see whether it is vaccine associated or not.
All right, thank you.
Thank you, Dr. Dlamini. Okay, then I'm not sure if there's anyone else with a hand up, so otherwise I'm going to continue with the questions here. So Manoko Mabocha wants to know, would people be tested for COVID first before receiving the vaccine? No, at the moment, everyone is in agreement that even if you've had it before, getting a new dose now would just be a booster and it is safe to take it. So AK Desai asked effectively the same question about how effective is the vaccine if the patient has antibodies. If okay, how long should we wait to give the vaccine after recuperation from COVID? The latest guidelines I've seen basically says that like any other vaccine, if the patient is acutely ill with a fever and anything else, you won't give them the flu shot while they have that. You won't give anyone the COVID shot while they have that. I think internationally, I'm not so sure. under correction, they say either give it a month or 3 months. I would need to check that specifically after you've been diagnosed with COVID. So it is, there should be a lag between diagnosed with COVID and getting a shot. Which vaccine would I personally want to get at this stage? At this stage, I will take anything, to be quite honest. I mean, I think a lot of people were initially worried about the AstraZeneca because the numbers are lower than Pfizer and Moderna. But I have to say, personally, I don't think it looks too bad. I think it's, I like the data they've added for the asymptomatic infection as well, because I think we would need to know if a vaccine is helpful with asymptomatic infection, and because that will help to curb the spread of the virus. And yeah, so I will take anything that anyone is willing to give to me. Possible long-term autoimmune disease for Zila Mun, she said. Not sure if that is a question, but we don't have data, and I haven't seen that anyone in any of the trials have mentioned that they, for example, had arthralgia or, you know, fever, prolonged fever or anything like that. So we will need to see, but I think it's not something to worry about at this stage. Can cancer patients receive the vaccine from Devendra Bika? I know who has recently sent out the guidelines. Under correction, I can't remember. A lot of the societies still say they will need to get more information. Some of the others say, yes, you can. So I will need to investigate that one and get back to you to see if there's anyone locally that says yes or no. But I mean, I think for the same reason, we do not know the effectiveness of the vaccine in severely immunocompromised patients due to any other reason. So that would still need to be determined. I think one of the societies said they can't theoretically think of a reason why not. But good question, but I would need to see exactly what everyone is saying. So is the vaccine contraindicated in pregnancy? Unfortunately, that has not been investigated. That is the big question always about the pregnant population. So at the moment, most societies are saying do not give it. Some of them are saying now. I think someone mentioned earlier this week, there's 1 or 2 groups that are saying give it, others say do not. So we still need to determine exactly where to go. So I think you would probably need to weigh up the pros and cons about giving a vaccine and potentially having a problem, or not giving the vaccine and actually getting COVID during pregnancy. That would need to be discussed, I think, individually with each patient to see what you want to do. Does the current information indicate at how long the immunity will last after both doses, or should one get an annual booster with time? Really interesting question. I mean, not enough time has elapsed, so we cannot say that, but it may be, I'm not sure if I'm going to say likely, but it is possible that COVID will end up being like flu, because it is an RNA virus that does mutate. So maybe you would need a booster maybe every year or every second year to boost your immunity, that if the virus continues to mutate, that you are then protected against the new variant. So answering about, yeah, so AstraZeneca for doctors, but the efficacy is very low. I mean, it is lower, it's better than nothing. And I think the efficacy data for sort of severe COVID is also a good thing to keep in mind. And that's the other thing I think to remember for a lot of vaccines. A lot of the vaccines, the efficacy data, for example, the rotavirus vaccine, with that one, we tell people, you know, it won't prevent you from ever getting rotavirus, but it will prevent you from getting severe rotavirus and ending up in hospital. So perhaps that is the way to frame and to think about COVID vaccines the same way we think about a flu vaccine as well. It's not 100% effective, but efficacy against preventing severe infection, you know, being admitted to hospital and potentially passing away, that is something I think that is good enough for me to take the AstraZeneca vaccine. Is the vaccine contraindicated if a patient had Guillain-Barre syndrome after Zostavax? That's from Crandall Sapphire. Once again, excellent question. I think that is one I would be hesitant to, because we're not too sure exactly what in the vaccine causes the Guillain-Barre, if it's one of the additives that is added to stabilize the vaccine or not. And I know, I mean, in that case, if one has developed Guillain-Barre after vaccine, one is hesitant to give it any other vaccine to that patient. So in this case, I would also perhaps discuss it with the patient and see, you know, weighing up pros and cons to see which side if they want to take a chance, or if they would rather say, you know what, it is the Swiss cheese model. I think if I can continue social distancing, wearing my mask, washing my hands, all of those things, maybe this specific patient would then have to be one of the ones that we need to protect via herd immunity because they can't take the vaccine themselves. Sorry, next question. Why is there such a vast difference in Pfizer and Moderna vaccine when they're both mRNA? They don't have a big difference. It was 94% of efficacy versus 95. So I don't think that is a big difference between the two. So what is the percentage of people needed to achieve herd immunity? So the epidemiologists and the sort of big data scientists sort of theorized with mathematical modeling that we need at least 67% of the population to be immune to get or to achieve what, I think what they want us to call now population immunity and not herd immunity. So how effective is it against the new variant? So that I've only had the data from the press releases from earlier this week. So it's not super the best, but we would need to sort of see exactly the data. We haven't seen anything specifically from Moderna and Pfizer. They've only done sort of data from sort of in vitro studies, and in that they said that it is, it should still work. More than that, I think, unfortunately, we won't be able to say. Would there be a need to defer periods for blood donors after receiving the vaccine? How long would it be? Excellent question. I mean, I think that would depend on the blood bank. If they put in an embargo on time with that, I mean, I don't think whenever I've gone to donate blood that they've ever said, no, it's too soon after my flu shot that I can't donate. I need to, I'm talking under correction. And theoretically, I cannot see that it will cause a problem anywhere. because there is either mRNA or replication deficient, vector virus or a subunit. I cannot see that it will cause any problems in the donor your blood is transfused, or not the donor, the recipient your blood is transfused into. Interval between the different phases. I presume that's the phases from the government's vaccine rollout. Unfortunately, I do not know exactly what that is. So why was the vaccine only tested in persons older than 16 or 18? Nothing for kiddies, even kids with comorbidities from Mershanadia. I think that's the same category as pregnant women or people with cancer. They've always first test in sort of the healthier population. And I think, people are always hesitant to try something out on a child or on a pregnant woman. It's always, I think, also difficult to get that sort of past ethics approval or not. But a few of the vaccine manufacturers, especially the ones that were approved earlier, Moderna and Pfizer, they have indicated that they are looking into pregnant women and children as well. So hopefully we will have a bit of data on that side. Okay. Are any of the vaccines egg-based like flu? No, not at all. They're not grown in egg cells. So another question about positive before, do you still need to be vaccinated? I've asked on that. So someone asked about giving hepatitis B immunizations and then how long to wait before giving Covishield. That is the problem, we don't know. I mean, if it is at all possible to wait at a month, I want to say, try that. If not possible, I mean, as I say, theoretically, I cannot see that there should be a contraindication. But the big thing, of course, is, patients shouldn't be acutely ill with a fever before they get any vaccine. Okay, Dr. Dlamini, I've answered that question. Is it you can't? Oh.
Hi.
Right, Bila, I just saw your message that you're not able to unmute yourself, but I think you have. Do you have a question? Mershon did ask again about people on immunosuppressant drugs, and is there a certain CD4 count in HIV patients where it's safe to vaccinate? Unfortunately, we don't have that data yet. But I mean, none of the vaccines are live attenuated vaccines. So, yeah. I mean, I don't think you would need to wait, let's say for a CD4 count to be above 200, but that is something that needs to be looked at. I'm hoping that when we see more data from the South African Novovax and Johnson & Johnson vaccine data, it may be indicative. They said medically stable HIV positive patients, but they don't say what the CD4 counts and percentages are in these patients. So we may be able to infer something from the data from that study. So keep us posted. Are the vaccines safe to give in patients with ulcerative colitis and Crohn's? Great question. I don't think that has been investigated. I can't think that they've specifically seen that, but I think I would look at those patients as anyone else who potentially has some immune dysregulation, and to see whether you you want to give it or not. I don't know. Unfortunately, I can't answer that one, but interesting. And I think that is the last of the chat questions. I can't see another one. So if anyone else has a verbal question, please go ahead.
Thank you very much, Dr. Maree. And well, I think you've covered quite a lot of questions, and I want to apologize for in case you got problems with the connections. There was power failure, so that's why she's not connected now. So we unfortunately have to take over. And I just want to thank you on behalf of Clinix Health Group and Health IQ Consulting for a very detailed and informative presentation. Answered many questions about vaccines. I think it was also good that you dealt with the conspiracy theories. Just over the last few weeks, and I think we've been bombarded with information about how unsafe the virus, the vaccines are. But I think we're glad, as you so clearly indicate, that safety is not an issue. Efficacy, I think, and also Astrozenica Oxford vaccine that we'll be getting in the next few days or weeks will be quite safe for all of us as healthcare workers to use. So thank you very much. And I think from Lancet, we are quite thankful that they gave us time and space to come and talk to us this evening.
Thank you.
Also thanks, thanks very much to all the participants who joined us at our first webinar since last year in 2020, and we still in the middle of the pandemic, and we'll be continuing with our webinars every Thursday between 6 and 7:30. We'll be announcing which topics will be covered in the next week. And so thanks to the marketing team from Clinix Health Group, and we quite appreciate your participation and assistance this evening, and also from Lancet. And as I said, from Health IQ Consulting and Clinix Health Group. And we know that one of our leaders in this webinar, Antonio Venturini, is not with us and he's not well. So we just wish him a speedy recovery and hope he will be joining us in the next few weeks. And so have a good evening. Hope to see you again next week. Thank you.
Thank you, everyone. Goodbye.
Bye-bye.